Great. Well, thank you so much for having me here. I'm looking forward to exchanging some ideas with all of you. As you said, if you want to find the papers, the data sets, any of that stuff, it's all here. So this is our academic site where I put things that we are fairly confident of. And then if you want to see some half-baked ideas on things that I'm thinking about, a little broader than the sort of peer-reviewed stuff, then that's all here. And so what I'm going to talk about today is this idea of bioelectricity as a kind of cognitive glue that is important across disciplines, from basal cognition and diverse intelligence all the way to very practical things like regenerative medicine. And I try in our group to really span the spectrum from fairly fundamental philosophical issues to very basic practical advances in biomedicine and engineering and things like that. And so when it's going well, that's what we do. We try to make a connection between some very deep ideas to very practical outcomes. And so the main points that I'm going to try to get across today are just a few. First of all, I'm going to introduce a framework that I call TAME, stands for Technological Approach to Mind Everywhere. But the bottom line is that these kind of philosophical questions about what minds are, what selves are, and what causes are, and so on, is really important. It's not just empty philosophy. It actually, if we do it right, it actually drives new discoveries, and it has done in our lab. I'm going to argue that navigation of problem spaces, which is in another word for intelligence, is a kind of fundamental invariant when we seek to recognize, create, and relate to very unconventional agents. And you'll see what I mean by that. I'm going to spend the majority of the time talking about anatomical control as an example. In other words, I'm going to treat the collective intelligence of cells as they navigate morphospace as an example of diverse intelligence. And I'm going to say that bioelectrical networks are the medium, the protocognitive medium of this collective intelligence, and thinking about it that way has really important practical implications for biomedicine. And then at the very end, I'm going to talk about synthetic engineering and this idea of really a very large option space of possible bodies and minds. So this is kind of fundamental to the idea of diverse intelligence, many of which do not have standard evolutionary backstories, which means we have to think about them as quite differently. And I probably won't get to do it in the talk itself, but if anybody wants to talk about it afterwards, we can talk about these synthetic constructs as an exploration tool for the latent space of possibilities. So let's start here. Let's broaden our ideas beyond the standard model systems that we all study. So this is a very famous painting. This is Adam naming the animals in the Garden of Eden. And there's a few interesting things here. One which is deeply wrong, and one which I think is deeply correct. What's wrong is that in this worldview, there are very strict boundaries between types of beings. So it's very clear what the different animals are. It's very clear that Adam is different from them. This is the idea that there are these natural kinds, and they have sharp boundaries between them, and that's that. That I'm going to try to dissolve today. The part that I think they got exactly right in this idea is that in the traditional story of Adam naming the animals, notice that it's really the first human, it's Adam, that has to name the animals. And when I say name, I mean the kind of the notion that when you name something, you really are discovering its true essence. You know, giving something a name is very powerful. It means you've understood something fundamental about what it is. And in this story, it's not God that names the animals. It's not the angels. None of them could have done it. It's on Adam to do it. First of all, because he's the only one that can do it, and second, because he's the one that has to end up living with them. And so it's on him to discover the fundamental nature of these things. And I think that's deeply correct, because as you'll see towards the end of the talk, we as humans are going to have to uncover the true natures of all kinds of unusual things. So our work is not done. It's not just about the standard set of familiar animals here. So so this idea that Adam is somehow a discrete different category was stretched already in two ways a long time ago. One by Darwin in the evolutionary sense, where we know that this modern standard human is actually just one of a whole continuum of very sort of smoothly transitioning forms. And the same thing was done in developmental biology, where it's very clear that, again, this is on a faster time scale, but it is basically a continuum of forms that gives rise to this, and in fact, some very different forms. And not only is there this sort of vertical continuum around this atom and whatever sort of agential glow he has about him that we think humans have all these special capacities, you might ask yourself what happens to those properties as you go down through the spectrum. But actually, it's a lot more complex than that, because there's also this kind of horizontal spectrum where, both by technological and biological modifications, we know that this is not a discrete category. There are all kinds of in-between cases that can be made. It's a very very plastic and continuous set of deformations. And in fact, now we know through bioengineering and synthetic morphology that every layer of organization can be either replaced or augmented by different scaled engineering artifacts or novel biology chimeras and so on. And so this already starts to tell us that we really need to understand a lot more widely than the standard things that evolution has given us. And so what I'm interested in are frameworks that allow us to relate to, and that means in certain senses that means predict and control, but in other cases it means having a deep ethical relationship with all sorts of unfamiliar beings. And so in addition to primates and maybe birds and octopus and things like that people study intelligence in, we need to be able to understand all kinds of colonial organisms and swarms and engineered new life forms, AI, whether embodied or purely software, and maybe someday even exobiological agents. And certainly not the first person to try for a framework like this. So this is Rosenbluth, Wiener, and Bigelow, who are trying to make a scale where you can try to understand how basically passive matter ends up modifying itself to the point where eventually you have these sort of very complex second order metacognitive capacities that humans have. So I would like a modern framework for putting all of these things in one in one domain. And very importantly, this can't just be philosophy. It has to move experimental work forward. It has to be practically useful. It can't just be something we argue about and sort of forget it. It has to have empirical consequences that can tell us if we're on the right track and why. And so one of the things that I think about really hard is this idea of an axis of persuadability, such that we can put along the spectrum some very simple machines, and if you want to change what they do, you're not going to convince them of anything. You're not going to reward and punish them. It's really hardware modification is your only tool. But then you get to something more interesting, and these are the things that cybernetics gives us. And so these might be the thermostats and other homeostatic agents, where actually, in addition to you could rewire them, but actually even better might be to reset their set point. And so you see here that as we go up this continuum, you don't really need to know quite as much about the low-level mechanisms, because they offer interesting interfaces which make life a lot easier for you. Like humans were training dogs and horses millennia before we knew any neuroscience. Why is that? Because these kind of systems offer this amazing learning interface where we can use rewards and punishments, really knowing very little about what's underneath, and get very complex effects. And then, of course, up here, you can, instead of trying to train a system into something, you maybe communicate cogent reasons and arguments, because you're dealing with that kind of system. So what changes along here is not only the capability of making very complex outcomes with very simple triggers, and not only how much you have to know about the mechanism of the system to get it to work, but also this idea of what tools do you need. So down here you have familiar sort of tools of physics and chemistry, and by the time you get here, you're into, I don't know, psychoanalysis or something, and then behavior science, and then cybernetics, and so on. So it's really a question of what tools are you going to apply. So one could ask the question, where do cellular collectives fit in? If you have a bunch of cells, well, the current assumption is that it's going to be somewhere here. Maybe some people are sort of working at it at this level. But the key thing in my framework that I think is important is that you can't just guess. You have to do experiments. You can't have feelings about it. You have to do experiments, which means using the different tools that belong to these different categories. And of course, there are more categories. And then you try it and you see how it works. And then you find out whether you're at the right level or not. And skewing low, which is what we're told in, for example, basic biology, that don't think about goals and reasons, think about chemistry and chemical processes and mechanics. Well, you can find out whether that gives you everything you want, or whether that leaves a lot on the table, which is what I think. So we have to start at the very beginning and remember that we came from a single cell, and so this is an unfertilized quiescent oocyte, and everybody would look at this and say, okay, well, that's a chemical machine. It obeys the laws of physics and chemistry, and that's pretty much that. But eventually we end up as something like this, which has complex the hopes and dreams and metacognitive capacities. And what's critical is that this is a slow, gradual process. And already you see the problem is people looking at this and they say, well, humans do all this incredible complicated stuff. These cells, they're just molecular machines. And once you've adopted that binary frame, you have a really hard time understanding what happens here, because developmental biology does not offer any lightning bolt halfway through that says, at this point, you've gone from physics to mind, okay? So we really have to absorb the lessons of development and understand that this is gradual. These binary categories that we paint onto things are really not helpful. Okay, so that's a little bit disturbing that we somehow kind of self-assembled from this. But at least we're a unified intelligence. Yeah, people think, okay, well, ants and termites and bird flocks and things like that, well, they're a kind of collective intelligence, but we're not like that. We're a real unified intelligence. And in fact, Descartes really liked the pineal gland because there being only one of them in the brain, he felt that the unified experience of being a human needs a central unified node in the brain to correspond to it. But if he had access to good microscopy, what he would have found is that, wait a minute, there isn't one of anything in the brain. What you see is actually a huge number of cells in this pineal gland, and inside each one of those cells, you actually see all of this stuff. So in fact, we are all collective intelligences. In fact, I think all intelligence is collective intelligence. We are all made of parts. This is the sort of thing that we're made of. This happens to be a free living organism called Lacrimaria, but you can see what's going on here in one cell. So there's no brain, there's no nervous system. This whole, this animal solves all of its metabolic, physiological, behavioral, and anatomical needs at the level of a single cell. No brain, no cell-to-cell communication, no stem cells, and so on. And so we really are starting to understand that all the magic and mystery here is in the scaling. You know, how do we get from the competencies of these kinds of things to the sorts of things that higher level animals can do? And I want to really hammer this concept of plasticity a little bit before we go on, because again, I think it really helps us focus on some problems that typically aren't talked about, because they really stretch our understanding of how cognition maps onto its physical embodiment. So here are caterpillars. Caterpillars live in a two-dimensional world. They crawl around and chew leaves, and they have a brain that's suitable for that. They need to turn into this. They need to turn into a butterfly, which is actually, so this is a kind of a soft-bodied robot, and it has a controller for that. This is a hard-bodied architecture. It flies. It lives in the three-dimensional world. Doesn't care about leaves. It likes nectar, and has a very different brain. In fact, to go from here to here during this process, the brain is largely dissolved. Most of the connections are broken. Most of the cells are killed off as the body reconstructs. And yet, actually, there's pretty good evidence that moths and butterflies remember things that their caterpillar larvae were trained on. So you can imagine what an amazing model this is. Not only, first of all, basic philosophy of mind, never mind what it's like to be a caterpillar. What's it like to be a caterpillar slowly changing into a butterfly, right? Where your cognition and the way you view your world is remodeled in real time. This is not evolutionary change. This is during the lifetime of the ancient. I mean, just incredible to think about it. Also, how are the memories stored such that they're stable to this massive refactoring of the medium? And so this kind of plasticity of mind and body. We have other examples. Here are planaria. These are flatworms. You'll hear more about them momentarily. But what happens here is that you can train them on something, cut off their heads. The tail sits there doing nothing for about 8 days until it regrows a brand new brain. And then you find out that they, in fact, still remember the original information. So that information is stored somewhere. We don't know where. It's imprinted onto the new brain as the new brain develops. And it leads to all kinds of interesting philosophical questions. When I chop this thing into pieces and they all have the original memory, this is a standard thought experiment for Philosophy 101. Which is the original you if we were to make a copy? And so you can make as many copies of these guys as you want. They also allow you to do another interesting question, investigations into another interesting question, which is what is the calculus of memory like within the body? So we see here that memories can be imprinted onto the new brain. But here, what we can do is we can train this animal on a particular task and take the head. we'll train this animal on a different task, and we'll take the tail, we'll fuse them together, which we can do in planaria, into a brand new organism. And now we ask the question, well, what happens? What's it like to be an organism that's composed of two different and perhaps inconsistent sets of memories, right? So just think about that idea of being a collective intelligence is really profound in terms of how you map your cognitive content onto the body, because we can do all these kinds of things. There is no theory at this point that even begins to make predictions about stuff like this. Okay, so what underlies all of these amazing properties, amazing capabilities of biology is this multi-scale competency architecture. It's the idea that not only are we tiered in terms of physical structure, so molecules, cells, tissues, organs, and swarms, and so on, but actually each of these levels has its own competencies. They solve problems in different spaces, in transcriptional spaces, in physiological spaces, in, of course, familiar anatomical three-dimensional behavioral space, and so on. And so having these different competencies suggests that maybe what evolution has done is use some of the same tricks in terms of navigating spaces, storing memories, making decisions, to climb up as agents get more and more complex. It climbs up from very simple metabolic concerns to physiological states to transcriptional, anatomical morphospace, then of course behavioral, and for some of us linguistic spaces. And you might ask, what are the invariants here, right? And we've done, I don't have time to talk about it today, but we've done Francesco, my group, has done very nice work looking at the same kind of navigation as also what you need to maintain the thread of a story when you're telling a linguistic narrative. So I think these kind of competencies are extremely powerful, and they can be rotated through different spaces. And I'm going to show you how that rotation works between this anatomical morphospace and the cognitive science style learning and memory. So, okay, so at this point, what I'd like to do is shift gears and talk about one very specific kind of example of an unconventional agent that lives and strives and suffers in its own space, which is anatomical morphospace, the space of all possible anatomical configurations. And we're going to talk about morphogenesis, so what you've heard of before as development, regeneration, cancer suppression, and so on. We're going to talk about this process of morphogenesis as the behavior of a collective intelligence. And there are many implications for evolution. I won't have time unless people ask me at the end to talk about that. And I'll mention briefly what I think this means for biomedicine. So let's talk about that. So here's Alan Turing. He's the kind of godfather of computer science. He was very interested in computation, thinking, problem solving, intelligence in machines, in unconventional substrates. And he was specifically, he worked out a scheme for problem-solving machines that exhibit intelligence through plasticity, aka reprogrammability. Now, interestingly enough, he also wrote this. He wrote a paper on the chemical basis of morphogenesis. He was interested in where the order of biological development comes from. Now, you might think that's kind of weird. Why would somebody that's interested in computing and math and intelligence, why would he be studying the self-organization of morphogenesis? And I think, although as far as I can tell, he didn't write about this, but I think if he had lived longer, biology would be much further ahead, because I think he was looking at a very fundamental invariance between the creation of the mind and the creation of the body. I think he understood that these processes were in some way very, very deeply isomorphic to each other. And I think that's a really critical insight. And I think he was really onto something. So where do anatomies come from? Here's a cross-section through a human torso. You see this incredible order. All the organs are in the right place. Everything's in the same, in the correct place next to each other, and so on. Where is this encoded? And you might think, as many people say, well, the genome. Of course, it's in the human genome. But we can read genomes now, and we know that none of this is directly in the genome. Genomes don't code for this stuff. Genomes code for proteins. They specify micro-level hardware that every cell gets to have. And so the outcome, this anatomy or the structure of this termite mound or the structure of the spider web, it's not directly in the genome any more than this structure or this shape is in the genome of those animals. We need to figure out, how do these cells know what to build? How do they know when to stop? If something is damaged, we need to know how to get them to rebuild it. And as engineers, we need to ask, well, what else is possible using that exact same hardware? What else can they build? And this is fundamentally Turing's notion of reprogrammability. What else can they do? And so as we think about these things, I like to keep my eye on kind of what our end goal is. And I think we can think about it this way. The fundamental end game for this field is what I call the anatomical compiler. So the idea is that someday you'll be able to sit down in front of a computer and you'll be able to draw the plant, animal, or organ that you want. It could be anything. It could be something standard, or it could be here, like in this case, this three-dimensional worm, three-headed rather worm. And the idea is that you should be able to draw anything and not worry about the molecular implementation. You should just say what the anatomy is that you want. And if we knew what we were doing, we would have a system that would compile this down into a set of stimuli that would be given to cells to get them to build exactly this. So the question is, what is that mapping? How do you convince a group of cells to build one structure versus another? And the reason we need this, of course, is that aside from understanding better evolution and development, most medical problems boil down to this: the inability to control what a group of cells builds. If we had good control over it, birth defects, traumatic injury, cancer, aging, degenerative disease, all these things would go away. If we had the ability to get a group of cells to build exactly what we want them to build. And what's critical here is that this is not some kind of 3D printer that places cells in the correct orientation. This is a communication device. This is something that translates between your anatomical goals and the anatomical goals and set points of the cells that gets them to build whatever you want them to build. Now, we're very far away from this. And the question is why? I mean, molecular genetics and biochemistry and so on have been around and making huge progress for many decades now. Why don't we have this? Why don't we have complete control over growth and form? I want to make a simple example. So here are baby axolotls, and these guys have little forelegs. At this stage, here are frog larvae or tadpoles. They do not have forelegs at this stage. In my group, we can make something called a frogolotl, which is just basically a chimera, a frog and axolotl embryonic cells. And now I ask a simple question. We have the axolotl genome. We have the frog genome. They've been sequenced. We know all the genetic information. Can anybody tell me whether the frogolotl is going to have legs or not? And say the answer is no. No one can say, because we simply do not have the ability to make those kind of predictions, because it is not a decision at the level of genes or even cells. It is the working out of a morphogenetic program, and we do not know how to predict the outcome of that software. Specifically, and we're very good at this kind of stuff, so manipulating how proteins and genes interact with each other, making these networks. But we're really a long way from understanding how large-scale anatomical decisions are made, such as, for example, should we form a leg? And if we do form a leg, is it going to be made of just the axolotl cells, or can they convince the frog cells to participate? I'll show you a little bit of data on that kind of thing. So here, this is where computer science or computer engineering was in the 40s and 50s. This is what it was like to program a computer. You literally had to interact with the hardware. You had to rewire the hardware all the way on the left of that spectrum that I showed you. And I think this is where molecular medicine is today. It's basically still stuck at this level because all of the exciting advances, so genome editing, CRISPR, pathway engineering, protein remodeling, all those kinds of things are at the molecular hardware level. We have barely begun to scratch the surface of the higher level intelligence that is in our substrate, the reprogrammability. And of course, the reason we have our information technology revolution is because we've moved from a focus on the hardware to understanding what the software can do. And this is why on your laptop, when you want to go from Microsoft Word to Photoshop, you don't get out your soldering iron and start rewiring, right? It's because the hardware is reprogrammable. And I'm going to argue that biology is that way too. So what is it that we're missing in staying focused on this level? What have we left on the table? What have we not exploited? And I think that's the competency of the material, aka intelligence. Now, what do I mean by this? Of course, there are many definitions. I'm not going to claim that this is the only definition, but it's a useful one to get us going here. So William James said that intelligence is the ability to reach the same goal by different means. So I like this definition a lot. It's cybernetic. It doesn't say anything about brains or whether you're evolved or engineered. It's pretty generic. It doesn't restrict you to one particular problem space. It just means that you can navigate to a particular region of your problem space with some competency when things change. Now, here's a very simple example of that kind of barrier, and it's in fact one that that James used, which is that if you have two magnets and they're separated by a piece of wood, the magnets are too dumb to go around and meet each other. Why? Because they would have to temporarily get further from each other. There's a gradient of there's an energy gradient that makes them try to stick together, and they do not have the capability of temporarily going around in order to reap gains later. So there's no delayed gratification. Romeo and Juliet, being kept apart by social barriers and other kinds of and physical barriers, do have the ability to do that kind of stuff. They have planning capacity. They have all kinds of tricks in order to do that. And then there's all sorts of stuff in between: autonomous vehicles and cells and mazes and rats and whatnot. So, so you can think about intelligence as the ability to navigate problem spaces when things change. Both things in the environment and things about yourself change. And we saw some drastic examples with these caterpillars and so on. So let's just talk about what kind of collective intelligence do cellular swarms deploy? Well, the first thing is that development, which reliably goes from a shape like this to a shape like that. So this is a journey in anatomical morphospace. Development is very reliable to a high degree of accuracy. However, it isn't hard coded, because if you cut early embryos of many species, if you cut them into pieces, you don't get half bodies, you get perfectly normal monozygotic twins. Each part recognizes the other part is missing, recreates what's necessary, it adapts. This is regulative development. And so you got this ability to reach that ensemble, that goal state of the normal target morphology from different starting positions, and in fact avoiding some local minima. So that's one example. In fact, that example of anatomical regulation doesn't stop for embryos. It goes on in terms of adults of animals that can regenerate. So here's an axolotl. These guys can regenerate their eyes, their jaws, their spinal cords, their limbs, ovaries, and so on. And so here what happens is if you amputate anywhere along the length of the limb, it will grow, and it will grow exactly what's necessary, no more, no less, and then it stops when it's done. That's the most amazing thing about regeneration, is that it knows when to stop. How does it know when to stop? Well, it stops when a correct salamander limb has been completed. So you can think about this as a kind of anatomical homeostasis. It has a set point. It has a particular location in the anatomical space that it tries to get to. If you drag it away from that point, it will do its best. It'll spend energy and it'll do its best to try to get back there. By the way, this is not just for worms and salamanders. Mammals can do it somewhat. So human liver is highly regenerative. Even the ancient Greeks knew that. I have no idea how they knew that, but they did. Deer regenerate antlers. So 1.5 centimeters of new bone per day. When and this is bone, vasculature, innervation, and skin, up to 1.5 centimeters per day when these things are growing. And then human children also regenerate their fingertips, and then they tend to lose that. Ability. So I've shown you one type of navigational competency, which is to be dragged away from the right location in anatomical space and find your way back by regenerating. But it gets even more interesting than that. So this is one of my favorite examples. This is the cross section through a kidney tubule in salamanders, in newts. And what you see here is that there's about 8 to 10 cells that are normally, and this is taken from a figure in 1945. What you see here is that there's normally 8 to 10 cells that work together to build this particular structure. Now, one thing you can do in newts, and you do this by increasing the amount of genetic material they have at the second cell division, is you can make polyploid newts, and if you make 4N, 6N, and so on, these larger ploidy animals, what you find out is that you still get a perfectly normal newt of the right size, and so that's already amazing, that the amount of DNA doesn't really wreck things. But what you see is that the cells get bigger to accommodate that new DNA, and in fact, when the cells are bigger, fewer of them get together to make the same size lumen. So that's the second amazing thing, is that it scales the number of cells to their new size. And the most amazing thing of all is that if you make the cells truly gigantic, then they will simply bend around themselves and leave an empty space in the middle. Now, this is a different mechanism. This is cell-to-cell communication. This is cytoskeletal bending. So that's a kind of top-down causation in the service of this large-scale anatomical outcome in that journey through morphospace. They're able to call on different, the cells are able to call on different molecular mechanisms to get the job done. Think about what this means in terms of the plasticity of what evolution has produced here. It hasn't produced a hardwired solution to a specific environment that these animals live in. It's produced a problem-solving machine. This creature cannot depend on having the right amount of DNA. It can't depend on having the right size cells. It can't depend on having the right number of cells. It has to do its thing with massive uncertainty, not just in the external environment, but also in its own parts. We don't have any technology that even comes close to this kind of thing because it doesn't overtrain on the evolutionary priors. It's able to solve problems in new ways. And we've barely begun to scratch the surface of how that works. So I hope you're seeing this, the idea of what I mean by the collective intelligence here. This is problem solving. This is solving problems you haven't seen before. And so let's just look at a very specific example. So this is this kind of anatomical homeostasis, where this is the story that we all learn in developmental biology class. This kind of feed forward open loop emergent process where genes interact with each other. They make proteins. The proteins do various things, and then eventually, bam, you've got this emergent complexity. But that's not the end of the story. If you try to deviate from here, and that could be through injury, it could be through mutations, teratogens, whatever, all kinds of processes, both genetic and biophysical, will kick in to try to get you back to where you need to go. It's an error minimization scheme or anatomical homeostasis. So now, on the one hand, this is sort of a familiar story. Biologists know all about feedback loops. Of course, the physiology understands homeostasis and so on. But there's something weird going on here because, first of all, typically in those feedback loops, the set point is a scalar. It's a single number. You know, it's a hunger level or temperature or pH or something like that. This is different. The set point here is a complex anatomical structure. So already it's strange because how do you store an anatomical structure in cell memory? And then the next thing is again, we're not really encouraged to think about goals in kind of mechanistic biology. We're encouraged to think about chemistry and things and physics and things like that. But the thing is that here's where the view kind of the lens that you put on this really matters, because if you do think that thinking in terms of goals is taboo, and so this, you know, basically throwing away all of cybernetics and control theory and things like that, you know, you really want to stick to the lower level, you really only have one choice. This is the only place you can make interventions, and that's really hard, because if you wanted to make changes up here, this is why we don't have an anatomical compiler, because if you want to make changes up here, working out backwards, well, what do I have to do back here, is an unsolvable inverse problem. It's very difficult in most cases. We have no idea how to do it in the general case. You know, you might ask yourself, well, if I wanted a threefold symmetry newt instead of bilateral symmetry, or if I wanted, you know, more eyes or legs or something, what genes would I change? It's very hard. But if you're willing to pay attention to this as a primitive goal directed process, as homeostatic process with a set point, you might ask, well, couldn't we just change the set point the way you do in your thermostat? Could we change the set point and let the system do what it does best, which is reduce the error to that set point, from the current state to that set point? So this is the hypothesis that we've been testing now for a couple of decades. And that leads to the most obvious question, which is that where's the set point? So this view makes a few strong predictions. You should be able to find the set point physically encoded somehow. You should be able to decode it. You should be able to rewrite it, and you should be able to make changes top down by rewriting this set point and leaving the underlying machinery completely intact and let it build. So we started thinking, okay, how would you encode a complex morphological set point? Well, the inspiration that we took was from neuroscience. In neuroscience, of course, we all have goal-directed behavior, and we store the set points that we're trying to reach. And it's done this way. There is the hardware, which are these electrical networks formed by cells which have ion channels that set voltage states. And these voltage states may or may not propagate across these electrical synapses, known as gap junctions. And the movement of these electrical states through the network is what underlies the software. So here you can see this in a living zebrafish brain. This group made an amazing video. where you can actually see the activity of a living fish brain. And there's this project of neural decoding, where neuroscientists hope that by decoding this physiology, they will be able to extract the cognitive content of the animal. So the commitment of neuroscience is that all of the memories, the preferences, the goals, the behavioral repertoires of the creature are stored in this real-time electrophysiological medium. So here we see this is a precedent. So here's how physics encodes goals, memories, and so on. Well, the amazing thing is that this is not just something that evolution discovered when brains came on the scene. This is a super ancient system. All cells in your body have ion channels. Most of them have the ability to form electrical synapses with each other. And so what we did was to try to explore the parallels between neuroscience and developmental biology. In fact, we've recently created an AI tool that makes this easier. You put in a neuroscience paper and it tells you what a hypothetical developmental biology version of that would look like. And it works very well because there are some really fundamental insights about why these things are similar. And so if you take a neuroscience paper and everywhere it says neuron, you just say cell, and everywhere it says millisecond, you say hour, you've got yourself a developmental biology paper. And that's because what I think evolution has done is pivoted some of the tricks that were used to control morphogenesis into the space of behavior and neurobiology. And so we can try the same kind of research program. We can look at the bioelectrics of the collective intelligence of the body, whereas here it's uncontroversial, right? Electrical networks are the cognitive glue that make you more than the sum of your neurons. That's just what it is. But could that be true here too? Could bioelectricity be the glue that allows individual cells to work together towards pretty grandiose goals in anatomical space, making organs? And so the idea is that whereas the electrical networks on the brain make computations relative to certain goals and expectations and control muscles to move your body through three-dimensional space, basically the exact same thing happens in the rest of your body, where these electrical networks are moving your cells through other spaces, for example, morphospace, right? So that's my claim. And that claim means that we ought to be able to appropriate all kinds of tools, both practical bench tools and concepts, from behavioral neuroscience into developmental biology. And we've done this in many ways. Everything from this kind of thing using voltage dye imaging. So we developed the first technology that uses voltage imaging fluorescent dyes to look at all the conversations that early cells are having with each other. So this is Danny Adams's work in my lab, looking at an early frog embryo where the collective is sorting out who's going to be left, who's going to be right, dorsal, ventral, and so on. And so we can do all of that. We can use various modeling and simulation tools. So this is Alexis Pytak's simulator called Betsy, which allows us to actually take some of these kinds of rules and ask what the bioelectric properties of the collectives are going to be. And I'm going to show you a couple of examples of these patterns. So here's a time lapse of an early frog embryo putting its face together, and the brightness value here is this voltage dye. It's a non-invasive way of probing the voltage, and so there's a lot going on, but you can see this is one frame from that video. And the reason I'm showing you this one frame is because here we can see this is a very easy, and some of these are much harder, but this is a very easy example of a bioelectric pre-pattern. It literally looks like a face. We call this the electric face. So here's the mouth, here's the animal's right eye, the left eye comes in slightly later. Here you've got the placodes. This before the genes are turned on to pattern this thing, you already can read out where the collective thinks all the organs are going to go, and what the plan is for gene expression and for anatomy. And I'm going to show you in a minute why we know that this is actually functionally determinative of what happens. But this is an endogenous pattern. It's absolutely required for normal development. If we mess with this pattern in any way, then the craniofacial patterning is altered. But here's a pathological pattern, and we'll get to this towards the end. But just to say that if we put in a human oncogene here, and it will begin to make a tumor, but even before it does that, already you can see that these cells have an aberrant bioelectrical pattern, and they're not going to participate in normal morphogenesis. They basically just decide they're amoebas, and the rest of the body is environment, and they metastasize and so on. And we'll talk about that story. But so you can imagine tracking these patterns for diagnostics in developmental for birth defects in development, and also for cancer screening, and when we're doing all of those things. Now, more importantly than just tracking these patterns, you need to be able to read and write them. And so what we do is we use the tools of neuroscience. Again, we do not do any kind of field applications. So there are no waves, fields, frequencies, electrodes, electromagnets, and nothing like that. What we're doing is using the native interface that these cells use exposed to each other to hack each other's behavior. And that means ion channels on their surface and gap junctions that connect them. So we can control, so this is some kind of tissue, not neural, and we can control now using optogenetics, using various drugs, different techniques like that. We can open and close the different ion channels, and we can control which cells talk to which other cells. Okay, so we can control the synaptic plasticity and the intrinsic plasticity of these cells, and then ask the question, what happens when we change the bioelectrical information that they have? So here's one of my favorite examples. Here's a tadpole, and you're seeing, here's the eye. So this is a side view. Here's the gut. The tail is back here. Here's the mouth down here. And so what we've done is inject some RNA that encodes a particular potassium channel. The reason that this works is because of that eye spot that you saw in that electric face. We can ask, what happens if we introduce that eye spot somewhere else? So instead of the face, we'll put it somewhere else. We'll put it on the gut. When you do that, these cells that normally participate in making this gut, they get the message that they are now supposed to make an eye, and that's exactly what they do. They make an eye. These eyes, if in cross-section, look, they have all the right layers, lens, retina, optic nerve, all that stuff. So here we learn a few lessons. First of all, the bioelectric pattern is in fact instructive. It's not a housekeeping parameter, which is what everybody thought around 2000 when we first started doing this work. And they said, well, if you change the channel expression, you're just going to get death, uninterpretable toxicity. So that's not the case. Bioelectricity is instructive. We can actually call up whole other organs, as you see here. Second, it's incredibly modular. So all we did was put in one potassium channel. We didn't really give all the information that's needed to build an eye. This is a subroutine call. This is a simple trigger that says build an eye here and offloads all of the complexity onto the rest of the system. The rest of the cells need to figure out how to do it. We don't do it, and as the ones triggering, and that of course is very okay, kind of a very desirable property from the perspective of regenerative medicine. The other thing that you see here is that by prompting these tissues in a different way, we discover novel competencies. Your developmental biology textbook will tell you that only the anterior ectoderm, so this stuff up here, is competent to make eye. And the reason it says that is because prior to this, people were using something called PAX6. It's a gene. It's called the master eye gene. And if you misexpress PAX6, yeah, in tadpoles, only this area is competent to make eye from PAX6. But if you go up a level and you prompt them with a bioelectric cue, you find out that no, actually, it's not a problem of competence. You can make it anywhere. We've seen eyes on tails and anywhere you want. And so this reminds us of an important thing in diverse intelligence research. When you make an estimate of the intelligence or the competency of some system, you're basically taking an IQ test yourself. Because if you miss out on what the system's actually capable of, well, then that's on you. You don't automatically know what things are capable of until you prompt them in the optimal fashion. And so that's a reminder to us to be humble about the competencies of the systems we study, because it often reflects our own limitations. And the final thing that I find super cool about this is that here's a cross section of a lens sitting out in the flank of a tadpole somewhere. What you'll notice is the blue cells are the ones that we actually injected with these potassium channels. These other cells were never modified by us. So there's a kind of a two-level instruction going on. We said to these cells, make an eye. These cells can tell that there's not enough of them to make a proper eye. And so they recruit all this other stuff. They recruit their neighbors to help them complete the task. And so this is something you see in other collective intelligences like ants and termites, right? When they find something, and the ones who found it will often recruit their buddies to help them move it or drag it back to the nest and so on. So all of these competencies, this modularity, the morphogenesis, the scaling, the recruitment, we don't have to bake any of that. We don't have to force any of that the way you would if you were trying to control all of this bottom up. These are all competencies of the medium, the agential medium in which we work, which is the cellular collectives. So with that trigger, that idea that you can trigger complex organ formation led to a regenerative medicine program where we try to regenerate legs. Frogs normally, unlike salamanders, do not regenerate legs, but here we discovered a cocktail of ion channel and other kinds of drugs that will trigger basically a grow whatever belongs here kind of message. They immediately turn on pro-regenerative genes. There's a blastema. There are some toes, a toenail by day 45, and the leg is both touch sensitive and motile. In our latest example, the treatment is 24 hours, and then the adult frog regrows its leg over a period of 1.5 years, during which time we don't touch it at all. It really is just communicating that early intent to the cells, and then you leave it alone and let them complete their journey. I have to do a disclosure here. So Dave Kaplan and I are co-founders of this company called Morphoceuticals Inc., because we're now hoping to take that into mammals and hopefully eventually into the clinic using a wearable bioreactor. So this is what David's lab pioneered, is this wearable bioreactor. Then we try to figure out the payload that goes in to try to convince the cells that they should go down the regeneration path and not the scarring path. Okay. So I want to switch now and talk for a couple of minutes about the planaria, because I want to really stress this idea of the bioelectric medium as the cognitive glue and really the substrate of intelligence, of memory in these systems. So planaria are kind of amazing because, as I said before, you can cut them into pieces. The record is something like 276 pieces, I believe. Each piece will regrow again to make a perfect little worm. And you might ask a question, how does it know how many heads, how does each piece know how many heads to have? In fact, when you cut them like this, the cells back here are going to make a tail, but the cells up here are going to make a head, but they were direct neighbors. They were sitting right next to each other before you cut them. How come they have such radically different anatomical fates? You know, it's not as if you can say that whatever cells were in this position are going to make a head, but it's because it's not true. So how do they make these decisions? And so we started asking this question: how does a fragment know how many heads to have? And we discovered this bioelectric circuit that actually controls the number of heads. And so here's what it looks like. You've got a one-headed planarian. You chop off the head and the tail. You take this middle fragment. Here you find that the anterior genes are up in the head like they should be. And 100% of the time, this large fragment is going to give you a normal one-headed worm. But in fact, what we can also do is produce these worms, and I'll show you how we did it, which again have the normal gene expression, anterior gene expression in the head and not the tail. But if you cut these guys, they become two-headed. And in fact, this is not Photoshop. These are real live animals. And the reason is that this animal here has a bioelectric pattern that looks like this, and it says one head, one tail. What we've done here is go in and mess with his bioelectric pattern to make it look like this, and it's kind of messy. The technology is still very much in development, but you can see quite clearly now this guy says two heads instead of one, and if you cut him, sure enough, that's what the cells make. Now here's the really critical point. This bioelectrical map is not a map of this two-headed animal. This is a readout of this one-headed animal. So what you see here is that the pattern to which you will regenerate in the future, the set point, I told you in the beginning that part of our research program is to find where the pattern is encoded. This is where now you're literally seeing now where it's encoded. This is the pattern to which I am going to build if I get injured in the future. Not only is it a memory, it is a counterfactual memory, because until you get injured, that isn't your anatomy. So your anatomy is perfectly normal, one-headed. Your internal representation, your self-model of what you should be is actually different. But that isn't going to, those two things don't come into alignment until you get injured. And at that point, the cells read the information, and sure enough, they build a two-headed worm. So a single worm body can have multiple, at least two, probably more, but at least two representations of what a correct worm is going to look like. Now, this is the precursor of the kind of mental time travel that we enjoy with our brains, the ability to remember or envision things that are not true right now. So we think this is a component of that basal cognition research program. And the reason I keep calling it a memory is because it has all the properties of memory. If I take this two-headed worm and I chop off the original head and I chop off this crazy ectopic secondary head, it doesn't go back to normal. I mean, it has a normal genome. Nothing's happened to its genome. It doesn't go back to the normal one-headed pattern. It continues to make these two-headed worms. So this is an example of inheritance of a somatic change. It is not done at the genetic level. It does not. It reveals that the number of heads is not something that's nailed down in the genome. The default is, so the genome builds a machine where the default pattern says one head, but if you change it, it'll keep. And so that's the key property of memory, is that it's stable, but it's rewritable. And here you can see this video. There's the first time I showed these data, somebody stood up and said, well, that's impossible. These animals can't exist. And so now I bring this video so you can see that's exactly what happens. And so it's very interesting to ask about the latent space of what else they can do. And in fact, we found that it's not just about head number, it's also about head shape. So if you cut off the head of this triangular headed animal and perturb the bioelectrical pattern, you can get flat heads like a P. felina, you can get round heads like an S. mediterranea. Without genetic change, this same hardware can visit other attractors in that morphospace that normally belongs to other species. They go there naturally. This one can be made to go there if you perturb that normal electrical machinery that controls how it navigates that space. And it's not just about the external shape of the head, but the shape of the brain and the distribution of stem cells becomes just like these other species. By the way, between 100 and 150 million years genetic distance between this, and they have no problem making those shapes. In fact, they can make much weirder shapes. So we've been able to make this crazy spiky thing, this three-dimensional kind of cylindrical thing, these hybrid forms. And the key is that it's not only human engineers that have figured this out, that you can actually push this, the hardware can be pushed into novel areas of morphospace. There are animals that exploit this as well. And so this, for example, this is a plant, this is a leaf from an oak tree. And you might think you know what an oak genome does. It produces this nice flat thing and it does it quite reliably. But if it's prompted by a wasp laying an egg on these things and putting out certain signals, then these cells can be hacked to produce something completely different. In the absence of this natural bioengineer, we would have no idea that these plant cells are actually capable of building something like this. So this is just, really important, this idea of plasticity and that morphospace and what the same hardware is willing to do with the right input signals. And so now, of course, what we're trying to do is to produce a full stack model where we can go from the expression of different channels and signaling factors and so on to tissue level bioelectrical dynamics, organ level patterns, and ultimately a logic of decision making where we can see large scale how cell collectives make decisions in the anatomical space, so that we can begin to have control, and ultimately going towards that idea of an anatomical compiler once we understand how native memories and goals are encoded in these bioelectrical states. And that means that when we look at the landscape of medicine in general, this is everything that's been emphasized so far in medicine. So it was all of these bottom-up interventions that basically target the hardware, but there's actually a lot that remains to be discovered. And I won't have time here, but you can look this up in these papers where we talk about the future of medicine and what it means when we really take advantage of that architecture that has information processing at every level, right? Instead of trying to force it but through the management of symptoms. And my claim is that future medicine is going to look a lot more like a kind of somatic psychiatry than it's going to look like chemistry. It's going to be all about managing the memories, the goals, the beliefs, the stress states, and so on of tissue and organ level agents. And I think that's going to be very powerful. So the last thing, just for a couple of minutes, I want to close and show you a couple of things, and then I'll stop. So one of the key things in this framework is this idea that the boundaries of the self, the boundary where agents end and the outside world begins, can be thought of as the size of their goals. So every kind of agent, whether engineered or natural or whatever, we can sort of plot the size of the goals that they're able to pursue on this kind of combination space-time diagram. And some creatures have little tiny cognitive light cones, and some are a little bigger, and then humans, can be huge. And the idea is that we are a collection of nested intelligences, all of which have goals in different spaces that interpenetrate. And the reason that this is useful in empirical application is this. Here's what evolution has done. Evolution has enabled, using bioelectric signaling and of course biochemical and biomechanical signaling, a radical scale up of these cognitive light cones. This creature has a cognitive light cone that's very small. It only cares about what's going on within that particular cell and sort of right around it. But together, this collective actually has a much bigger cognitive light cone that allows it to maintain the goal of having a limb. And if you deviate from that goal, it will get right back and try to implement it again. So these goals are huge. That's what evolution has done during multicellularity. Now, that process has a failure mode, and the failure mode is cancer. So when individual cells detach from that electrical network such that they can no longer participate in this high-level memory of a goal state in another space, they basically devolve to being amoebas again. And this is glioblastoma in vitro. And they just treat the rest of the body as external environment. And the reason to think about it this way is that it actually suggests a novel kind of therapeutic, because instead of trying to kill these cells, you might ask, well, what if we forcibly reconnect them to the electrical network? And that's what we've done here, where you put in really nasty human oncogenes like KRAS and things like that. And then you also put in an ion channel that forces these cells to be in electrical communication. So they are now once again bound to that network, which thinks about very lofty goals such as building nice organs and skin and muscle and so on, instead of the metabolic and proliferative goals of single cells. And sure enough, here's the oncoprotein. It's very strongly expressed. In fact, it's all over the place. This is the same animal, and there's no tumor. And that's because it's not the genetics that drives, it's the physiology / behavior that drives. And this is something that we're developing as a therapeutic. And so the very last thing I want to show you is how far you can push this notion of a latent morphospace. I keep using the word goal in the cybernetic sense as these patterns in different spaces to which the system will try to reach. And one question is, where do they come from? Where do these anatomical goals come from? And the typical answer is evolution. It comes from eons of selection for specific outcomes. And so we wanted to establish a model to ask where novel goals might come from. So this is kind of synthetic morphology. And again, I'll do a disclosure because Josh Bongard and I he's at the University of Vermont. We have a company around some of this stuff called Fauna Systems. And so Doug Blackiston and my group did this work. So basically, we take some cells from a frog embryo, we dissociate them, we liberate them from the rest of the body, and we ask, what are they going to do? Well, they could do many things. They could have died, they could have spread out and went away from each other, they could have made a nice flat monolayer, they could have done nothing. Instead, what they do is they come together and they make this. We call it a Xenobot. Xenopus laevis is the name of the frog, and we think it's a biorobotics platform, so Xenobot. And what it's doing is it's using the little cilia, the little hairs on their outer surface, which are normally used to propel mucus down the edge of the animal. It's using them to row against the medium. And so they're self-motile. They can go in circles. They can patrol back and forth like this. They have various collective behaviors here. Here's one running down, swimming down a maze. So it takes the corner without bumping into the opposite wall. It spontaneously turns around for some reason that we don't understand and goes back. So they have a range of behaviors. Remember, there's no neurons here. It's just skin. These are just skin cells having a new life outside the embryo, and they have novel capabilities. One of them is kinematic replication. So if you provide them with loose cells, these white things are loose skin cells. What you find is that the motion that they do ends up collecting these skin cells into little piles. In fact, they compact the little piles, both working together and singly. They compact the little piles. And because they're working in an agential material, like we were when we made Xenobots, otherwise this doesn't work, this ends up making the next generation of Xenobots, because guess what these piles do? They mature, they become the next generation of bots, and they continue to do exactly the same thing, and they build the next generation and the next, and so on. So you start to see these kind of things, and you realize that again, the standard outcomes that we see in development are a tiny point in the option space of what's possible given that hardware, and that the frog genome, in fact, yeah, typically this is what it makes, and there's a developmental sequence, and then there's some tadpoles with certain behaviors, but it can also make this. It can make this Xenobot within a couple months. It undergoes this transition. I have no idea what it's becoming. It's got its own sort of sequence, and then they have different behaviors here. And the important idea here is that while we can talk about all of these things having been shaped by selection, there's never been any Xenobots. There's never been selection to be a good Xenobot. And so all of this is an exploration of that latent space by the same hardware that has not been specifically selected for in this context. I mean, of course, the cells were part of the evolutionary stream of frogs, but there's never been any Xenobots. And we don't know what their cognitive properties are, by the way. This is actively being investigated right now. Do they have memories? What are their preferences, and so on? And so the very last thing I'm going to show you is, in case you get the feeling that this is some weird amphibian thing, or, well, it's embryonic. We know embryonic cells are plastic. We know amphibian embryos have these animal caps that are ciliated. Maybe this is just some kind of frog thing. Well, I'll show you this. This is maybe the first time I'm showing these things in a public talk, because the paper just came out. And this is the work of Gizem Gomushkaya from my group. This is so you look at this little creature and you ask, what is this thing? Where did it come from? Did we get this out of a pond somewhere? You know, some kind of simple life form. Could you guess the species of this? And I could tell you that, well, we know what the genome is. The genome is 100% Homo sapiens. This is we call these anthrobots. And what happens is that cells, ciliated cells from the trachea and the lung epithelium, put through a specific process, end up making something that is again a self-organizing kind of motile construct. Here they are. They move around. They have all kinds of interesting behaviors. One of the amazing things that we discovered is that if you put them on a scratch made in a sheet of human neurons, then a bunch of them that have gotten together, we call this a superbot. So these anthrobots can be made to come together. They land in this scratch, and what they'll do in the middle is they'll make the nerves knit together. If you lift them up afterwards, you see that underneath you had this thing going on. And so they have this latent capability for neural repair. This was basically the first thing we checked for. Who knows what else they do? So you know, it would have been really hard to know what your tracheal cells that sit there quietly for decades in your body, they sit still, if liberated and allowed to kind of refactor their multicellularity, what behaviors, what capabilities. I think we're just scratching the surface. And again, this is 100% standard human, no genomic editing, no weird transgenes, no drugs, nothing. This is the latent capabilities of your own body cells to heal you. So okay, so just to end here, I think that one thing that's really clear in biology, because of the plasticity and the ability of bioengineering to interact with the software of life, is that all of the diversity that Darwin was so impressed with when he said endless forms most beautiful are basically a tiny corner of, like, all the default N of 1 evolutionary lineages on Earth are a tiny corner of this option space of any combination of evolved material, designed, engineered material, and software is some kind of an agent. And we already have various kinds of hybrids and cyborgs and so on, and this is just going to explode. And in the next coming decades, we are all going to be living in a very different world with a wide diversity of bodies and minds all around us. And we need to get started on figuring out how we're going to relate to them. And the old categories of, were you natural or made in a factory, or what are you made of? Do you look like me? Do you have a frontal cortex like I do? All of those kinds of old criteria are going to be completely useless. So we need to start to really understand diverse intelligence, so that we can address not only the AI problems that are facing us, but a much wider set of beings that we're going to be living in, hopefully in some kind of ethical synthbiosis. So if anybody's interested in more details, here are some papers that dig into to some of the questions that I've really just glossed over. Here, I want to thank all the people that have done the work. So here are the postdocs and the grad students, the really amazing people in my group who do all the experiments. We have many collaborators. Here's our funders that have supported the various stories that I've told you about. Again, the disclosures. There are three companies that support some of our work, and most of all to the model systems, because really that's where all the information and all the hard work comes from. It's the animal model systems. So I'll stop here and take questions. Thank you so much.